Hi, my name is Lena True, and along with my partners Hana Kawamura, Katrina Lay, and Taylor Redmond, we investigated filtering runoff with fungi mycelium, or mushrooms. So, in the urbanized world, paved roads and non-porous surfaces leads to the collection of runoff that can contain industrial waste and synthetic chemicals. This is a hazard to human health and the environment. A possible cure is the use of fungi mycelium, which has the properties to metabolize hydrocarbons and filter out these toxins. Our study investigates the use of Oystritis pleurotus, or more commonly known as the oyster mushroom, and we specifically looked at the way different substrates or growing materials for this fungi would affect the efficiency of fungal filters in water.
To conduct our experiment, we made 7 groups with different mixtures of substrates, as seen in Figure 1. We placed these into separate burlap bags. Then, we placed the bags into individual containers to let them grow for the next 4 weeks, during which we recorded qualitative data. When it came to testing, we all tilted the containers to an angle and poured the synthetic runoff that we made over the course of 3 hours to simulate a stream. After waiting for 24 hours, we recorded the water quality with water testing strips.
So, most simply put, our results show that the test group using 50% straw and 50% bark chips performed the best and had the best water quality levels. As seen in our data, here's a chart showing the quantitative levels of how well each test group performed, with the larger score being worse, farther away from the water quality standards. You can see that the straw and bark performed the best and the coffee and straw performed the worst.
With the results in our experiment, we could say that the hypothesis was not supported by the data as indicated in the graph. Some of the weaknesses in our experiment was the fact that the substrate groups had to be grown separately amongst the members, leading to a difference in environment showing effective results. However, some strengths was that we could make our own synthetic runoff, so we can know what was in it and what quantities, and the before and after of the runoff after the filtering. When applied to a real-world environment in small-scale projects such as a slow-moving stream, wetlands, and other bodies of water, we can see that fungi would be an effective filter due to the fact that there needs to be not any pre-treatment to the filtered water, and this would be an advantage in filter efficiency. Further research on this project, we would include more water quality tests throughout the testing time so as to see what fungi has as an impact on the runoff, having a constant stream of water through the substrate bags as to mimic a real-life environment, and having a longer period of testing time to see more results and to refine filters.
In conclusion, our hypothesis was rejected. However, it does suggest that fungi can be used for water filtration. Real-life application with this data can include a plan for filtering water in smaller-sized bodies of water or local waterways. For acknowledgments, we would like to thank Mrs. Abraham and Mrs. Roberts for providing us with some of the materials necessary to execute this project. Thank you!